Welcome to today's lecture dealing with membrane structure. This will be the first lecture in a series of lectures dealing specifically with the cell structure itself. And with the cell membrane, what we are going to do is work from the outside in. But before we do that, I want to talk just a little bit about the fact that when we discuss organisms in general, they can either be single-celled, such as most prokaryotes and protists, and we're going to talk about some of those characteristics in just a minute, or they can be multicelled, such as plants, animals, and most fungi. This is, again, a cell biology course, so we're going to focus on the fundamental unit of life, which is in fact the cell, but just keep in mind that there are single-celled organisms and multicellular organisms. In our first lecture, one of the topics that I discussed briefly was how it is that we as scientists deal with the diversity of life. And the way that we typically deal with any type of diversity is to begin to categorize components so that it's a lot more manageable as far as a data set is concerned. The first main categorization that deals with the diversity of life focuses on the type of cell in question. There are two broad categories of cells. That is, those cells that are known as prokaryotes or prokaryotic cells and eukaryotes or eukaryotic cells. And as we discussed in that first lecture, there are in fact three domains of life. Bacteria and archaea are all single-celled organisms that are prokaryotic cell types. And the domain eukarya are made up of either single-celled or multicellular organisms, but the cell types are known as eukaryotic cells. This includes protists, which are single-celled eukaryotic organisms, and plants, fungi, and animals, things we might be more familiar with. Now, before I highlight the two different categories of cell types, whether it's prokaryote or eukaryote, I like to talk about cells in general. No matter what type of cell we're talking about, there are three characteristics or three components that you find in every single cell type. The first one is that every single cell is bound by a thin plasma membrane. Today's lecture is going to focus on that membrane structure, and we're going to hopefully understand the membrane structure in a good amount of detail. But every single cell has a membrane that is some type of boundary of the cell. It is a phospholipid bilayer, which again we're going to talk about in more detail today. But what this does is act as a selective barrier. It allows passage of oxygen, nutrients, and waste. So oxygen and nutrients in, waste out. But again, this is a boundary or a border of the cell overall, and every single cell has it. Now, in addition to every single cell having a plasma membrane or a cellular membrane, every single cell will possess DNA at some point in its life cycle. DNA, as we talked about in the last lecture series, is in fact known as the genetic material. It contains all of the instructions necessary for a cell to function. The third component that every single cell has, or feature that every single cell has, are structures known as ribosomes. Ribosomes are small particles that carry out protein synthesis and are essential for life as we know it. These tiny structures within the cell again, are the components that synthesize proteins that the cell will need to survive. So again, three features that every single cell is going to have. It's going to have a cellular or plasma membrane that acts as a boundary for the cell. It's going to possess DNA at some point in its life cycle, and every single cell will have ribosomes to synthesize proteins necessary to carry out life. In addition to that, I want to mention one additional component, and that's the term at the bottom of the slide, which is cytoplasm. The cytoplasm refers to the entire contents of the cell. So everything you find inside the cell is referred to as the cytoplasm. And as we continue on with our next set of lectures, we will talk about all of the contents that we can find inside a typical cell. Now, before we move on, I want to highlight some basic differences between typical prokaryotic cells and eukaryotic cells. The term prokaryote literally translates to no kernel. What this means is that the cells that were first identified in this category seem to lack a central structure that other cell types actually had. This kernel refers to the nucleus, which is found in eukaryotic cells. The nucleus is a structure that houses the genetic material and protects the genetic material, and it is the defining feature or characteristic of eukaryotic cells. The term eukaryote literally translates to true kernel. So again, historically, the two cell types were recognized as having a kernel, which is really the nucleus, and those are eukaryotic cells, and lacking a kernel or a nucleus, and those were prokaryotic cells. Now, there are a number of additional differences between the two categories, but that's the first defining feature or characteristic difference between prokaryotic cells and eukaryotic cells. Let's go ahead and take a closer look at prokaryotic cells first. Prokaryotes are typically smaller than eukaryotic cells, and they lack internal structures that are surrounded by membranes. And we'll talk about those structures when we talk about eukaryotes in general on the next slide. As I mentioned just a minute ago, these particular cells lack a nucleus, and a nucleus is a membrane-bound structure that houses DNA. Most prokaryotic cells will also have a rigid cell wall, and this allows for structure and protection of the prokaryotic cell. Now, we won't talk about prokaryotic cells in much more detail than this one particular slide in this class. You could take a microbiology class where the focus would definitely be more on the prokaryotic cell type. So for now, let's talk about the things that are common in a typical prokaryotic cell. As I mentioned on the last slide, every single cell is going to have a plasma membrane or a cell membrane, and this is a membrane that encloses the cytoplasm and acts as the boundary or the border, dividing the interior environment from the exterior environment. As I mentioned on the last slide, every single cell type will also have ribosomes, which are the structures that synthesize proteins necessary to carry out life. I also mentioned on the last slide that every single cell will have DNA at some point in its life cycle. Prokaryotic cells do in fact have DNA. They do not have that specialized structure known as the nucleus to house the DNA, but they do have a region of the cell where the DNA is found, and this is referred to as the nucleoid region. In addition, like I mentioned just a minute ago, prokaryotic cells will have a cell wall, which is a rigid structure outside of the plasma membrane that provides structure and, more importantly, support to these single-celled organisms. Now, some prokaryotic cells may also have something known as a flagella, which is plural, and flagellum, which is singular. And this is a locomotor appendage, and that's what you see in this figure that looks sort of like a long tail protruding from the prokaryotic cell. The flagellum, again singular, is a locomotor appendage, and this allows for the prokaryotic cell to move in its environment. Some prokaryotic cells will also possess a capsule, which is an outer polysaccharide coating. This usually is a defense component that prevents engulfing of the prokaryotic cell, so other organisms can't necessarily attach or attack this cell if it has an exterior capsule. Lastly, not all, but some prokaryotic cells will have pili, and pili is plural, pillus is singular, and that in this figure looks like those little hairs that are sticking off of the prokaryotic cell. These are structures that are oftentimes used for attachment, either attachment to other cells or attachment to surfaces in their environment. The other broad category of cell type is a eukaryotic cell. And when we talk about eukaryotic cells, we oftentimes subdivide them into an animal cell and a plant cell. And these again are generalized figures that we're looking at on this particular slide. However, eukaryotic cells are fundamentally similar to one another. The most important defining feature or characteristic of a eukaryotic cell is in this figure, that purple structure, that is the nucleus. The nucleus is a membrane-bound structure, meaning it has its own membrane associated with it, and this is where the DNA is found or housed, and it allows for protection of that genetic material. In addition to that, eukaryotic cells also have organelles. Organelles are structures with specialized function, and we are going to talk extensively about those in our next lecture. So the plasma membrane or the cell membrane is what separates the living cell from its non-living surroundings. It is the boundary between the interior environment of the cell and the exterior environment outside of the cell. It is a barrier between the cell and its environment. Nutrients need to be able to move in, waste needs to be able to move out. So it is actually quite a dynamic structure that allows for movement of molecules into and out of the cell. It is extremely complex. It's involved in cell signaling mechanisms. It's involved in import and export, like I just mentioned, allowing molecules to come in or molecules to exit. The cell membrane does typically play a role in movement or motility of a given cell, and it also is extremely important for expansion and cell growth. At the center of the cell membrane is a structure or molecule known as a phospholipid. So in our last lecture, we talked about lipids, and then we discussed 3 categories of lipids. Phospholipids were in fact one of them, and we talked briefly about the phospholipid structure, and we're going to definitely talk about it in more detail today. It is specifically the structure of the phospholipid that allows for the phospholipid bilayer to form. As you can see in panel A typical phospholipid bilayer looks something like that. Bilayer just means there are 2 layers of phospholipids that make up the membrane structure, and it's not by mistake that these phospholipids are oriented in a specific way. If you recall, when we talked about phospholipids, we discussed the fact that a portion of the phospholipid is hydrophobic and a portion of the phospholipid is hydrophilic. And it is this dual nature of the molecule that allows for the formation of the phospholipid bilayer. The hydrophobic fatty acid tails tuck in away from the water, and the hydrophilic phosphate group, which is also referred to as the head of the phospholipid, is facing aqueous environments, both externally and internally. Now, we know that the membrane is actually much more complex than just a simple phospholipid bilayer. In panel B, you have an example of what the membrane structure looks like, and it is referred to as a fluid mosaic model, and we'll talk about that in just a minute. What I want to highlight, though, is that not only do you have a phospholipid bilayer, but in a typical membrane, you will also have proteins embedded in that phospholipid bilayer. I'd like to include this particular slide because it has 3 different figures that represent the phospholipid bilayer. The figure on the left-hand corner represents a more complex view of the phospholipid bilayer. You can see the phospholipid bilayer. You can see the proteins embedded in that phospholipid bilayer. You can also see that there are structural components outside of the cell. This is referred to as the extracellular matrix or extracellular fluid. And then on the inside of the cell, just inside, you can also see that there are additional structural components that attach to the proteins that help with cellular structure overall. And we will talk about some of these components a little bit later in today's lecture. In the right-hand corner, you have a color-enhanced electron micrograph image of a phospholipid bilayer. In the orange is the fatty acid tails, and in the red is the hydrophilic heads. And in the blue represents the aqueous environment overall. And this is a really nice image because it gives you a feel for what that structure really looks like rather than the nice, neat cartoon figures that we usually focus on. And then lastly, in the bottom right-hand corner, there are 3 different panels with this particular figure. In panel A, you are looking at an electron micrograph image of the plasma membrane. Panel B gives you a 2-dimensional schematic view with the proteins you can see again embedded in that phospholipid bilayer. And then panel C gives us this sort of 3-dimensional view of what the phospholipid bilayer looks like. Let's now take a closer look at phospholipid structure. If you recall from the last lecture, a phospholipid is made up of a glycerol molecule and 2 fatty acid chains, as well as a phosphate group. In the far left, you can see a more structural view of what this molecule looks like. In the yellow is meant to represent the fatty acid tails. In the gray box is the glycerol as well as the phosphate group, and we'll talk about some additional groupings in just a minute. This molecule is said to be amphipathic. That term amphipathic means that it is a molecule that contains both hydrophobic and hydrophilic regions. And we've already discussed this and reviewed it in today's lecture, but phospholipids are in fact amphipathic. The fatty acid tails are hydrocarbons that are dominated by non-polar covalent bonds, which makes that portion of the molecule hydrophobic. The phosphate group in particular, in the phospho part of phospholipid, has a net negative charge associated with it. And because it has a negative charge associated with it, it is going to very readily or easily interact with water, which is again a polar molecule. So you have this molecule that has both hydrophilic and hydrophobic portions. In panel B, or in the center of the figure, you can see more of the space-filling model associated with that structure. And then lastly, what you've already seen in some of the figures that we've looked at is a more idealized cartoon version of a phospholipid that just highlights the hydrophilic head and the hydrophobic tails. In this figure, we actually are looking at a molecule known as phosphatidylcholine, and this is the number 1 or most common type of phospholipid structure. It is known as phosphatidylcholine because in addition to the basic phospholipid structure, there is a covalent linkage of a choline molecule to the phosphate group, and you can see that in panel A as well as in panel B with the space-filling model. This figure also gives you an example of some additional molecules that can be found in a phospholipid bilayer. You have on the left-hand side another phospholipid known as phosphatidylserine. It has that same structure that we just focused on in the last slide, but in the blue is a serine molecule, and that's attached to the phosphate group. So it just makes this a little different type of phospholipid. On the far right, you have a molecule that again is very similar in structure to a phospholipid. This is known as a glycolipid, and that's because it does in fact have a carbohydrate that is linked to the structure, and that's what you see in the 6-membered ring structure at the very top of that figure. This particular molecule is known as galactocerebroside, and it is, again, another component that you can find in a phospholipid bilayer. Lastly is the molecule in the center, which is cholesterol. Now, cholesterol, we talked about a little bit at the tail end of last lecture as well. Cholesterol is a molecule that is not a phospholipid, but a steroid. And steroids or this particular steroid, cholesterol, is also an important part of membrane structure. Now, the common features of all three of these molecules is that they are amphipathic in nature as well, that there is a component that is hydrophilic and a component that is hydrophobic, which allows them to interact with the typical phospholipid membrane structure we just discussed. Based off of the general understanding of the components found in a typical membrane, in 1935, Hugh Davison and James Donnelly proposed what's referred to as the sandwich model. What they proposed in the sandwich model is seen in the top left-hand panel or figure, specifically panel A. In panel A, you have the phospholipid bilayer that is known to form based off of biochemical studies with phospholipid structure. And the proteins, because proteins were known to be found in membrane structure as well, were thought to be sandwiched on either side of the phospholipid bilayer. So this is again referred to as the sandwich model. And so in the sandwich model, you would have the proteins that would sort of be like the bread in a sandwich on either side. And in the middle would be your meat and your cheese, or the phospholipid bilayer. Now, in the 1950s, electron microscopy work demonstrated that there did seem to be three distinct components or layers to a phospholipid bilayer. And in the 1960s, this was accepted even for internal membranes. But it turns out that there were some problems with this particular model. Not all membranes looked alike when they're analyzed under the electron microscope. There are some additional membranes of structures inside of cells that, when analyzed biochemically, have a greater protein percentage compared to other membrane structures. So different percentages of lipids and different percentages of phospholipids, and typically a lot more proteins. In addition, membrane proteins have also been determined to be amphipathic. And if the protein itself is amphipathic, meaning that there's a portion that is hydrophobic and a portion that is hydrophilic, it's very hard to reconcile that with the sandwich model. In 1972, the work of Singer and Nicholson proposed a different type of model, and this is the readily accepted model for membrane structure today. What they proposed was that the proteins that are part of a membrane structure are inserted into the bilayer, and this is referred to as the fluid mosaic model. Now, on the right-hand side is some experimental evidence that helped them support this particular hypothesis, and in this study, what was done is referred to as a freeze fracture experiment. What you can see is that you have a cell that's essentially frozen, deep frozen in liquid nitrogen, and then a very small and precise knife would crack open the cell right at the membrane. When you do this, you peel apart the two halves of the phospholipid bilayer. And what they found when they analyzed this was definitely that there were proteins that are embedded in this phospholipid bilayer rather than having that typical sandwich model that was proposed many years before that. So again, what they proposed is something known as the fluid mosaic model. The mosaic component comes from the proteins that are embedded in the phospholipid bilayer. And the fluid component comes from a set of experiments that I'm going to talk about next. In the bottom left-hand panel, you have an experiment that was done. This is a very simplified view of the experiment, but what you have are two different cell types, a mouse cell and a human cell. The mouse cell proteins are tagged with a dye or a marker that allows us to identify the mouse cell proteins, and that's in the pink in this particular figure. And the human cell proteins are tagged with a marker or a dye that allows us to identify the human cell proteins, in this figure purple. What was done in this experiment is the two cells were forced to fuse together to form a hybrid cell. And after about an hour, what they did was analyze the location of the proteins. And what they found was that the proteins sort of mixed and moved throughout this new hybrid cell structure rather than staying in a fixed position like you would expect if there's no movement associated with the membrane. So this is the fluid part of the fluid mosaic model, and that the membranes are fluid, meaning that there's constant motion or movement associated with those membranes. Let's talk a little bit now more about the fluid portion of this fluid mosaic model. When we use the term fluid, what we mean is something that's not static. It's not fixed in place. It's not rigid. There's movement associated with it. It is dynamic. It's specifically these hydrophobic interactions and hydrophilic interactions that hold the phospholipids together in the phospholipid bilayer. But because each individual phospholipid is not linked or bound to the neighboring phospholipid, that is one feature or characteristic that allows for this movement or fluidity. If we look at the phospholipids a little more closely, we can see that movement of phospholipids allows for fluidity. Movement, like you see in panel A, can occur in lots of different ways. You can have lateral movement, basically side to side in position. You can have what's referred to as a flip-flop, which is actually rare, but where you have a phospholipid that was in one layer of the bilayer move to the other layer. You can have rotation along an axis. The degree of saturation of the fatty acid chains or tails also plays a really big role in membrane fluidity. Recall when we talk about fatty acids, they can either be saturated, which means they have the maximum number of hydrogens bound to carbons possible. This again gives a very straight structure to the fatty acid tails. However, if you have at least some degree of unsaturation, that means that you don't have the maximum number of hydrogens bound to carbons, which really means that there's at least one position where there's a double covalent bond. If you recall when we talked about the structure, so unsaturated fatty acids again have bends or kinks in their chain and they're not nice and straight. What this means for the molecular structure and the behavior of the molecule is quite different. When you have straight linear structures in your fatty acid tails, this allows for the phospholipids to pack really tightly up close to one another. When you have bends or kinks in the chains, it allows for a little more space between neighboring phospholipids. The best analogy I can give for this is if we were in the classroom and I asked all of us to stand up and put our arms straight down at our sides. I could push us all into a corner. We'd be kind of uncomfortable, but I could push us all really, really close to one another and fit into a compact space. If one person that was in the very corner needed to move, it would be very difficult for that individual to do if we were all packed into the tight corner. If, however, we all had our arms up at arm's length, it would be very difficult to compact us into a tight space. There'd be a lot more room in between individuals, hence a lot more movement. If one person, the one in the corner, needed to get out, there'd be a little more space and maneuverability that could take place to allow that to occur. So again, the degree of saturation does in fact play a really big role in membrane fluidity. Lastly, the presence or absence of cholesterol, specifically in animal cells, is a big contributor to changes in fluidity. More rigid, less permeable in moderate temperatures, So when you have cholesterol present, it makes the membranes more rigid, less permeable, and this is usually in moderate temperatures. In lower temperatures, the presence of cholesterol actually hinders solidification. So it depends upon the temperature and how much cholesterol is present, what exactly it's doing. But think about cholesterol as being sort of like a roadblock. If there's a lot of cholesterol, it again is going to inhibit movement overall. Okay, so lastly, what I want to talk about is the mosaic portion of that fluid mosaic model. The mosaic portion refers to the fact that proteins are embedded into the phospholipid bilayer. As you can imagine, there are lots of different types of proteins that exist that are considered membrane-associated proteins. And just like we discussed at the very first lecture, when there's a lot of information, what do scientists do? They start to categorize them to sort of make sense or rhyme or reason associated with a large set of information. And that's exactly what we've done with membrane proteins, is that we can categorize them into different components based on their function. One category of membrane-associated protein are anchor proteins. Now, anchor proteins, their job is to basically maintain the cellular shape. If you see in this particular diagram at the very bottom the word cytoskeleton, we haven't talked about that yet, but we will. And the cytoskeleton is an internal network that adds structure and support to the cell. Anchor proteins are going to be proteins that are going to anchor the membrane to this cytoskeletal component. In addition, these anchor proteins can also attach to extracellular components as well. So one category is referred to as an anchor protein or anchor proteins. Another category refers to acting as a binding site for chemical messages. Basically, this is involved in cell signaling. There can be receiving of information from other cells or sending of information inside the cell, and these proteins, again, because they sit in the cell membrane at the boundary of the cell, can receive these signals externally and cause things to happen internally inside the cell. So molecules that act as cell signaling molecules are also one large category. Another category of proteins are those that act as enzymes or play a role in enzymatic activity. There are many examples where proteins are associated with the membrane and are an ordered team in a stepwise process that needs to occur. So oftentimes it makes sense to have these enzymes lined up one right after the other to basically make sort of an assembly line for the biosynthetic process involved. So another category for membrane-associated proteins is to act as an enzyme or enzymatic activity. Yet another category of membrane-associated proteins are those that are involved with transport. We talked at the very beginning of this lecture that it's very important to be able to move molecules into the cell and out of the cell, and there is a whole family of proteins that act in this manner to allow for transport of molecules into and out of the cell to take place. Yet another category involves cell-to-cell linkage or intercellular joining. In this case, you have membrane-associated proteins from two cells that interact with one another to allow for cell junctions to take place. This is very important for multicellular organisms, as you can imagine, to have these cells basically stick together or stay together in the organism itself. Lastly, we can have proteins that are involved in recognition, cell-to-cell recognition more specifically. This is to allow for cells to be able to recognize one another, and there's going to be a protein that is going to have some type of signaling molecule that is going to be recognized by the second cell and its protein that's associated with its membrane. So again, as you can see, there are lots of different categories associated with these membrane proteins. We talked about 6 broad categories. There are others, but these are the 6 main categories overall.